Hello students, welcome to ATPH Academy. Well, I am your Sir Krishnendu Sir, and today we're gonna learn about biomolecules. This is the chapter of class 11. Okay, it is one of the most important and interesting chapter of class 11, and I hope you will enjoy this class very much. And after completing this class, you can answer all the questions of your school or of your syllabus. Okay, so biomolecules, what is biomolecules? Our body is our body is consist of different type of elements. Okay, and these elements together form different types of molecule. Okay, you know that our body is composed of different type of element. What? Different type of element, and these elements accumulately form molecules, and these molecules inside the biological body is known as biomolecules. Okay, what are these biomolecules? So let's move to the next slide to to answer this, to get the answer of this question. Okay, here biomolecules, this is biomolecules. The biomolecules present inside our body can be classified into two groups, that is organic and inorganic. In organic, there are biomacromolecules and biomicromolecules. This is biomicromolecules and this is biomacromolecules. Okay, inside the biomicromolecules, we have amino acid, we have sugar, we have nitrogen base, we have lipid and fatty acid, glycerol. And in case of biomacromolecules, we have protein, polysaccharide, nucleic acid, that means DNA and RNA, right? Then, in case of inorganic molecules, there are two types of inorganic biomolecules: inorganic element and inorganic compounds. In case of inorganic element, there are calcium, magnesium, sodium, potassium, etc. In case of inorganic compounds, there are sulfate, phosphate, NaCl, that is sodium chloride, CaCO3, that is calcium carbonate, etc. Okay, so this is the basic classification of biomolecules, that those are present inside our body. Okay, among these we're gonna read detailsly, or we're gonna learn detailsly about these biomolecules. I mean organic biomolecules, that is amino acid, sugar, nitrogen base, lipid, fatty acid, glycerol or protein, polysaccharide, nucleic acid, that is DNA, RNA. Okay, so let's learn. At first, amino acid. What is amino acid? Okay, here in this the name amino acid, here is acid and amino. That means it is an acid which composed of an amino group, one or more than one amino group if present in an acid, that is known as amino acid. Okay, so amino acids are the building blocks of protein. That what it means. Okay, if you observe the protein present in our body, suppose consider this is a protein, this is a protein. Okay, this is a protein. You know that proteins is a biomacromolecule. I already discussed about that. This is a protein. Suppose this is a protein, and in these proteins, each and every bits is known as amino acid, right? That means protein is a polymer. Protein is a polymer, and amino acid is a monomer. Is it clear to everyone? Okay, so that's why Here it is written as amino acids are the building blocks of proteins. Okay, they are organic compounds containing both an amino group, that is NH2. NH2 is the amino group. The formula of amino group is NH2, and a carboxyl group. The formula of carboxyl group is COOH, along with a specific side chain that is considered as R group. Okay, so what contain amino acid? Amino acid contain an amino group and carboxyl group, and a side chain or a R group. Okay, so in general structure of amino acid, that is the formula of amino acid, general formula is the RCH NH2COOH. Amino group, that is NH2, carboxyl group, that is COOH, hydrogen atom, that is H, and a side chain R group. Okay, what actually looks like amino acid? Here I gonna draw a general structure of an amino acid. Suppose this is a carbon atom, and we know that carbon atom has four valency. Okay, four valency. In this valency, suppose an hydrogen atom is present. In this valency, suppose a carboxyl group is present, and in this valency, suppose a NH2 or amino group is present, and in this group a side chain or R group is present. So this is a general structure of an amino acid. Here a carbon molecules, and it have four valency. In one valency there is hydrogen atom. In the next valency there is carboxyl group. In the next valency there is amino group, and the fourth valency have a side chain or it is called R group. Okay, so that's why the general formula of amino acid consider as RCH whole NH2 COOH. Is it clear? Well, now the type of amino acid. What are the types of amino acid? There are so many types of amino acid on the basis of different things. Okay, on the basis of availability of amino acid inside our body, it is classified into two types. Availability inside our body on the basis of on the basis of availability inside our body. Amino acid is classified into two groups, that is essential amino acid. They are essential amino acid and non-essential amino acid. What is essential amino acid? Well, in which type of things are essential in front of you? I mean, in your point of view, which things are very essential for you? Those things which you actually don't get every time, that means those things you don't get every time, these are or those are the essential things for you. So these amino acids, these essential amino acids cannot be synthesized by the body. That means body cannot synthesize essential amino acid inside the body. So if we want to get this essential amino acid, we have to obtain this from our diet. Okay, so obtained from diet. These are known as essential amino acid. For example, valine, leucine, and lysine. These are the essential amino acids. That means these amino acids are not produced inside our body from its own. We have to consume this amino acid through our diet. Okay, so these are known as essential amino acids. Now, which are non-essential amino acids? These amino acids can be synthesized in our body. I mean, whenever I needed this type of amino acid, our body can synthesize it from their own. So they are non-essential amino acid. For example, glycine, alanine, serine, these are non-essential amino acids. I hope that this slide is clear to everyone. Now, move to the next slide. Now, classification of amino acid on the basis of side chain or R group. We already saw the R group. We already discussed about it, the R group, where R group is present in the amino acid. Now, on the basis of side chains, amino acid can be classified into non-polar or hydrophobic amino acid, polar or hydrophilic amino acid, acidic amino acid, basic amino acid. Okay, so hydrophobic amino acids examples is glycine, alanine, valine. Polar and hydrophilic amino acids example is serine, threonine, glutamine. Acidic amino acids example is aspartic acid, glutamic acid, and basic amino acids example is lysine, arginine, histidine. Don't worry, we will discuss every single amino acids in details in the next slide. Okay, so don't worry, don't be panic, just listen to these classes, listen to this class very carefully and enjoy this class. Okay, now, on the basis of nature, amino acids can be classified into neutral amino acid, acidic amino acid, or basic amino acid. That means which type of amino acid it is. If it is neutral, if it is acidic, or if it is basic. Okay, so these are the classification of amino acid. Okay, so let's move to the next slide. Nextly, these are the amino acids. How many amino acids? Mainly 20 amino acids are present inside our body. Okay, 20 amino acids, but nowadays there are 22 amino acids. Two more amino acids are newly added in this list. Here, those two amino acids which are newly added is not present in this slide. That is the exception. You don't have to be remember right now. Okay, you just have to be remember this 20 amino acid. This is glycine. Look, this is glycine. This is alanine. This is valine. This is leucine. This is methionine. This is isoleucine. These are non-polar. These are non-polar aliphatic R group. Non-polar plus aliphatic. Aliphatic means they are linear. The R group have linear structure, not circular or ring structure. In chemistry, you will read what is aliphatic, what is aromatic. I can only give you an example right now that aliphatic chains are linear chains and aromatics actually not chains. Aromatics are ring structure. Okay, like these are aromatic. Okay, because they have ring structure. But in case of here R group, R group, R group, R group, R group, R group, you can see that R group, these R group. These are in linear structure. So these are aliphatic R group, and obviously these are non-polar. What the non-polar means actually? Non-polar means they does not attract the water. Okay, they does not attract water, or they does not have the affinity with water. Non-polar means non-polar means here I will write this non-polar do not have the affinity with water. Okay, what polar means? Polar means they have affinity with water. Okay, so these are non-polar. Okay, now these are polar R groups: serine, threonine, cysteine. proline, asparagine, glutamine. These are polar R groups. That means they have the affinity with water. Okay, these are positively charged R group. It means these amino acids R group have positive ion. Okay, positively charged R group like lysine, arginine, histidine. These are negatively charged R group, that is aspartate and glutamate, and these are non-polar but aromatic R group. These are non-polar aliphatic R group, but these are non-polar but aromatic R group like phenylalanine, tyrosine, and tryptophan. Okay, now you can clearly observe the R group in the different color, like this is an R group, this is a R group, this is a R group. Okay, these are the R group of amino acid. Now each and every R group have its specific name. We will discuss this name now. Like alanine. Alanine have methyl R group. This group name is methyl. Okay, in exam it can be asked to you that what types of R group is present in alanine amino acid. You should give your answer like methyl R group. Okay. In valine, there is isopropyl R group. Isopropyl R group. in leucine, in leucine, there is isobutyl R group. Isobutyl R group. In methionine, there is sulfur containing R group. What? Sulfur containing R group. Okay, in isoleucine, that is sec butyl R group. That is sec butyl R group. Isoleucine sec butyl R group. Okay, in serine, in serine hydroxymethyl R group. This is hydroxymethyl R group. Okay, in threonine and cysteine, that is sulfhydryl R group. In cysteine, sulfhydryl R group, and in case of proline, that is pyrrolidine ring. This is not aromatic structure. You will confuse. I think you are confused in this structure, but this structure is not the aromatic structure. The R group is present here is this. Okay, this is the R group. Okay, but it is not a ring structure. I mean, it is not aromatic structure. It is an allophatic structure. But obviously, this is ring structure, but not aromatic. Okay, so that's why they are written as pyrrolidine ring in case of proline. In case of asparagine, this is amide group. Amide group. In case of glutamine, this is also an amide group. This is also an amide group. In case of lysine, this is amino group. This is amino group. This group is amino group. Okay, amino group's charge is positive, so this is positive amino acid. And in case of arginine, that is guanidino group. This group is known as guanidino group. Guanidino group. Okay, here also NH2 plus is present, so this is also positively charged. In histidine, that is imidazole group. In histidine, that is imidazole group. Imidazole group, here NH plus is present, so this is positively charged amino acid. In case of negatively charged as amino acid, here aspartate have carboxyl group. Carboxyl group have negative charge. Carboxyl group have negative charge. In case of glutamate, here also carboxyl group is present. So that's why this aspartate and glutamate is negatively charged amino acid. Next, these are aliphatic group, I mean aromatic group, phenol group. Okay, phenol group. Phenylalanine have benzyl group or phenol group. Phenol group, this is the phenol group. This is the benzyl group. Okay, this is actually benzyl group. That's why the name is phenylalanine. Next, tyrosine. Next, tyrosine. Tyrosine have phenyl ring with hydroxyl group. Phenyl ring with hydroxyl group. Okay, and next tryptophan have indole group. Tryptophan have indole group. Among these tryptophan this arginine, this histidine, this lysine, this aspartate, this glutamine, asparagine, proline, cysteine, serine, alanine are so much important. Okay, are so much important. Clear? I hope this slide is clear to everyone. If it is not clear, then observe or enjoy or replay the video again. It will be very clear to you. Okay. Next, move to the next slide. Okay. Next, the concept is deuterium. This is one of the most important and interesting concepts of this chapter. Deuterium. What is deuterium? In aqueous solution, amino acid exists as deuterium or dipolar ion. In aqueous solution, amino acid exists as deuter ionic form or dipolar ionic form, where the carboxyl group loses a proton, that is COOH, COOH to COO minus. A proton, that is H plus, is lost, and the amino group gain one proton, that is from NH2 to NH3 plus. Okay, NH2 to NH3 plus. Clear? Now deuter ion and its isoelectric pH. What is deuter ion? Deuter ion is the state of an amino acid in which state both amino acid contain both a negative charge and a positive charge. That means its main charge, its total charge is zero. Is zero. Zero charge. That particular state of the amino acid is known as deuter ionic stage. Okay. So if I draw an amino acid, look, I draw an amino acid. Here R group, here if COO minus, and here if NH3 plus, then this is known as in deuter ionic stage. Why? Because here one minus charge and one plus charge both are present in this amino acid, so the net charge is actually zero. So in this case, this amino acid is present in a deuter ionic stage. Okay. Okay, so if I give an example to you, like this is an amino acid. Okay, so this is a positively charged amino acid because here one positive charge is present but no negative charge. So the charge of this amino acid is positive. So the charge of this amino acid is positive. Okay, next, this amino acid at first, this amino acid at first donate this hydrogen, at first donate this hydrogen, okay, this carboxyl group hydrogen. After donating this hydrogen, it will convert it into this form. Here charge is zero because one negative charge, one positive charge is present. Okay, so this is the actually deuter ionic stage. Deuter ion. Okay, next, next, this hydrogen one proton is donated from amino group. Okay, Next, one proton is donated from amino group. So here the structure is like this: NH2 and R. You can observe the charge here. The charge of this amino acid is negatively charged here. There are no positively charged. Okay. So this is the conversion of positive amino acid to deuterionic stage and then negative amino acid. Okay. This is here one proton is removed. One proton is removed. Here second proton is removed. Here second proton is removed. Okay, and this particular deuterium is formed in a particular pH form, in a particular pH, and this particular pH is known as isoelectric pH. What? Isoelectric isoelectric pH. Okay, so in isoelectric pH, amino acid transform into the deuter ionic stage. Is it clear? So what is isoelectric pH? In which pH amino acid are present in the deuter ionic stage is known as isoelectric pH. It's very simple. Now, if I give you a positive amino acid, okay, a positive amino acid, here plus one charge, here minus one charge. Now suppose here I draw an amino acid which have plus 2 charge. Here one plus charge, and in R group, suppose in R group there are another plus charge. You consider it that R group have an amino group, that means positive charge. So here the charge is plus 2. The charge is plus 2. Okay. From here at first this hydrogen, I mean this proton is removed from this amino acid. This proton is removed from this amino acid. Here C double O minus, here NH3 plus, here R plus. The charge of this amino acid is plus 1. Okay, plus 1. Charge of this amino acid is plus 1. Plus 1. Okay. Now, which proton removed from this amino acid? This proton, R group's proton removed from this amino acid. So after removing this, the structure of amino acid is just like this: C double O minus, NH3 plus, and R. Here the charge of this amino acid is plus 1 and minus 1 is equal to 0. Okay, that means this is the deuterionic stage. This is the stage of deuterion. Okay, next, once more H plus or proton can be removed from here. Okay, so H plus is removed from here, and after removal of this H plus, the structure of amino acid should be this. The charge of this amino acid is minus 1. Clear? So this is the deuter ionic stage. This is the deuter ionic stage. And in this stage, the pH is isoelectric pH. If I consider a negative amino acid, which an amino acid which charge is -2. Okay, an amino acid with charge is -2. Then similarly, this type of patterns will be drawn. Okay, I think this slide is clear to everyone. If it is not clear, then please repeat this video. Repeat this particular section of this video. It is very easy. Just listen carefully what I am trying to say. Okay, let's move to the next slide. Now here look isoelectric pH. The isoelectric pH or pI of an amino acid is the pH at which the molecule carries no net electrical charge. That means in this pH the charge of an amino acid is 0. Okay, no net electrical charge. At this point the amino acid exists as a Zwitter ion. Okay, with its positive and negative charges balanced. The key points about isoelectric pH is deuterium form. At the isoelectric pH, the carboxylic group, that is COOH, is deprotonated to COO minus. That means carboxylic group যেটা ছিল COOH, সে তার proton, actually carboxylic group remove its proton, that is deprotonated, that means remove each proton, deprotonated and form COO minus, and the amino group NH2 is protonated to NH3 plus. Amino group NH2, okay, NH2, it is protonated, it should be protonated to NH3 plus. Okay, the net charge of the amino acid is 0. Okay, 0. This is known as isoelectric pH. Clear? Next, protein. We already discussed about that amino acids are the monomer, and from these amino acids actually protein is formed. Protein is a polymeric state of amino acid. Okay, protein is a polymeric state of amino acid. Protein is formed through amino acid, one by one amino acid joined together to form the protein. Okay, so let's detailsly learn about protein. So protein are the complex organic molecule that play a fundamental role in the structure and functioning of living organism. Okay, inside our body, proteins have so many amounts of role. Okay, so these are known as they play a fundamental and structural and functioning of living organism. So many amounts of roles protein actually do inside our body. Okay, they are often referred to as the building blocks of life. Protein is known as building blocks of life because they are essential for growth, repair, and maintenance of cells and tissue. They are very crucial for growth, repair, and maintenance of cells and tissue. Okay, what is the definition of protein? So proteins are the polymers. Okay, proteins are polymers. I already discussed this. Proteins are polymers. Proteins are polymers of amino acids linked by peptide bond. Okay, so inside the protein, one important bond is known as peptide bond present. More except peptide bond, there are so many amounts of other bonds also present, but peptide bond is one of the most important bond present in protein. Peptide bond folded into specific structure to perform a variety of biological functions. Formation of protein. How protein is formed? By peptide bond. How protein is formed? By peptide bond protein is formed. I will show you in the next slide. Don't worry how peptide bond actually form. Okay. A peptide bond is a covalent chemical bond. In chemistry, you will read there are so many different types of bonds are present, electrostatic bond, covalent bond, dipole-dipole bond, hydrogen bond, so many bonds are present. But peptide bond is a covalent chemical bond that forms between two amino acids during protein synthesis. It is the fundamental linkage in proteins connecting amino acids to form polypeptides and ultimately functional proteins. Okay, so these peptide bonds are the fundamental linkage in protein. Fundamental linkage, I mean there are so many other bonds in the protein, but peptide bonds are the fundamental linkage in the protein that connect two amino acid to form a polypeptides and ultimately functional protein. Okay, process of the formation of peptide bond, a condensation reaction also called dehydration or synthesis occur. That means a reaction where water is removed, H2O removed, H2O removed. Okay, so that is condensation reaction. A molecules of water H2O is released. The carbon atom of the carboxyl group forms a covalent bond with the nitrogen atom of the amino group, and the bond has the structure of CONH. Okay, so let's make a peptide bond. Okay, here I will make a peptide bond in between two amino acid. Clear? So suppose this is amino acid 1. This is the structure of amino acid. We already discussed about that. C double O H. C double bond O O H. This is C double O H, and here N H 2. NH2, and here R 1. That is amino acid 1. Amino acid 1. Okay. It will link to the amino acid 2. Okay, here amino acid 2, C double bond O OH N H2. This is NH2 or amino group, and this is R2, that is side chain. This is amino acid 2. Okay. Now peptide bond will form in between this OH, this OH, and this H. So ultimately OH plus H, what are actually removed. That's why this reaction is known as decondensation reaction. And after removing, the structure that is developed in between these two amino acid is like this. N double H NH2, this is R1, and here N H C H R2 C double O OH. Okay, this is amino acid 1, this is amino acid 1, and this is amino acid 2. And the peptide bond here present is this. CONH bond. CONH bond. CONH bond. This is known as peptide bond. Is it clear? By this way, peptide bond is formed in between two amino acids. Okay, now here amino acid 2, if amino acid 3, 4, 5 are present, in between those amino acid also peptide bonds are present. I mean peptide bonds are formed like this way. Okay, so I think how peptide bond is formed, it is clear to everyone. Now move to the structure of protein. There are four levels of protein structure. These are primary structure, next secondary structure, next tertiary structure, next quaternary structure. Primary, secondary, tertiary, quaternary. Four types of or four levels of protein structure are present. First, primary structure. Primary structure are a linear sequence of amino acids. Here you can see this is one amino acid, this is second amino acid, this is third amino acid, this is fourth amino acid, this is fifth, this is sixth, this is seventh, eighth, ninth, 10th, 11th, 12th, 13th, 14th, 15th, 16th. That means what? Linear arrangement of amino acid are present in primary structure of protein. So primary structure of protein is in linear structure. What type of structure? Linear structure. Linear structure. Linear structure. Okay. Next, secondary structure. Secondary structure of protein. Local folding patterns, for example, alpha helix, beta sheet stabilized by hydrogen bond. So, in primary structure, only peptide bonds are present. In primary structure, only peptide bonds are present. But in secondary structure, there are peptide bonds and hydrogen bonds. I mean, if I write primary structure, only peptide bond, only peptide bond, only peptide bond. But in secondary structure, there are two bonds, peptide bond and hydrogen bond. Okay, peptide bond and hydrogen bond. Now, what type of secondary structure are present? Secondary structure is of two types. That is alpha helix, and beta sheet. Alpha helix are of two types. One, right-handed alpha helix. Next is left-handed alpha helix. Beta sheet is of two types. One is parallel and next is anti-parallel. Anti-parallel. Okay. So right-handed alpha helix picture is this type. This is right-handed alpha helix and this is left-handed alpha helix. Parallel structure like this is parallel beta sheet, and anti-parallel, this is anti-parallel beta sheet. Clear? These are the secondary structure of protein. So the most important point of this secondary structure is here both peptide bonds and hydrogen bonds are present. Okay, always remember these bonds, in secondary structure. Okay, let's move to the next slide. Next, tertiary structure. Three-dimensional of folding of protein due to interaction among R group. This is the highest structural level of peptide. Okay, this is the highest structural levels of peptide or polypeptide, that is three-dimensional folding of protein. So if I draw a tertiary structure, this is like this. Suppose here alpha helix is present. Next here beta plated sheet is present. Next here alpha helix is again present. Next here beta plated sheet is present. Next here alpha helix is again present. ঠিক আছে, next again beta plated sheet is present. Next alpha helix is again present. So this is the actual figure of tertiary structure. Okay, so in tertiary structure different types of bonds are present. Obviously peptide bonds, hydrogen bonds, these are present, and along with this van der Waals forces, dipole-dipole interaction, these are also present. So in tertiary structure, what type of bonds are present? Firstly, peptide bond. Firstly, peptide bonds are present. Secondly, hydrogen bond are present. Thirdly, van der Waals forces. Fourthly, electrostatic forces. Fifthly, dipole forces. These are present. Okay. One of the example of tertiary structure of protein is myoglobin. This actually helps in O2 transport in muscle. 153 amino acids, it contains 153 amino acids. This is the tertiary structure, is the green cell. This is an example of tertiary structure of protein. Remember, if this tertiary structure is the highest structural level of a polypeptide. Look, in this, I draw this polypeptide chain. This is only single polypeptide chain. This is only single polypeptide chains. There are no more than one polypeptide chains. Okay, there are not present more than one polypeptide chains. So we can say that tertiary structure is the highest structural level of a polypeptide, that means single polypeptide. Okay, this is the highest level. In next slide, quaternary structure. In case of quaternary structure, association of multiple polypeptide chain, that means association of multiple tertiary structure actually is togetherly or accumulately known as quaternary structure. Like hemoglobin. Hemoglobin, obviously listen the term hemoglobin. This is an example of quaternary protein. This is the functional structure of a protein. Like here you can see the picture. This is one polypeptide chain. This is alpha 2. This is one polypeptide chain. This is beta alpha 1. This is one polypeptide chain, that is beta 1. This is another polypeptide chain, that is beta 2. Here four polypeptide chains is present. Four polypeptide chains togetherly form the quaternary structure. So if I draw quaternary structure here, look, suppose this is an alpha helix, this is a beta plated sheet, This is again a alpha helix. This is a beta plated sheet. This is again a alpha helix. This is one polypeptide chain. I will draw next polypeptide chain in this form. This is beta plated sheet. This is alpha helix. This is beta plated sheet. This is alpha helix. This is beta plated sheet. Okay. Next, this is beta plated sheet. This is alpha helix. This is beta plated sheet. This is alpha helix. Okay. So togetherly this will known as togetherly this is known as quaternary structure of protein. Quaternary structure of protein. Quaternary structure of protein. In quaternary structure, how many amount of bonds are present? First of all, peptide bond, peptide bond. Second, hydrogen bond. Third, van der Waal force. Fourth, electrostatic force. Electrostatic force. Fifth, dipole dipole force. And sixth, this is the most important, disulfide bond. Disulfide bond. This bond is only present in quaternary structure. This is disulfide bond is only present in quaternary structure. ঠিক আছে? I mean it is right. So this is the structure of protein. I hope you understand the structure of protein very well. Let's move to the next slide. This is the function of protein. What are the function of protein? At first, structural role, provides support, that is collagen in connective tissue. Enzymatic role. Enzymatic role, protein have enzymatic role, catalyze biochemical reaction, for example amylase, protease, these are protein and enzymes. Transport, carry molecules, for example hemoglobin transport oxygen. Protein helps in regulatory function, hormone and regulatory roles, for example insulin, growth hormone. Defense, protein form antibodies that protect against the pathogen. And protein have storage, that store essential molecules like ferritin, store iron. Okay, these are the few roles or functions of protein inside our body. Okay, in today's class I hold in this protein portion. In the next class I will continue from sugar. Here we will know the more interesting once more interesting topic of this chapter, the structure of sugar. Here different types of sugar is present. Then after sugar we will discuss fats, that is fatty acid. After fatty acid we will discuss lipid nucleoside, that means DNA, RNA. One of the most interesting part of this chapter, DNA, RNA. After that this chapter will be finished. And obviously there are enzymes also. We will also discuss enzymes here, but today's class is finished. I will hold this class right now. I'll stop this class right now. Okay, so I hope today's class is very useful to you, and you came to know different things from today's class. And thank you. If you want to get admission in my offline classes, then obviously quickly get admitted into my offline classes. We will provide so many facilities to you, like practical like digital classroom, so many exams in the year, and everyday MCQ test in the Telegram. We will provide these all benefits to you if you enroll with us in offline mode or in online mode, whatever you want. Okay, so thank you. Have a great day. We will meet you soon. I will meet you to the next video. Okay, goodbye.